Our approach makes it possible to control neural network formation using magnetic manipulations. This is an effective tool for in vitro studies of networks and offers novel therapeutic direction for biointerfacing devices.
With this technique, we can control both cells' location and growth at the micron scale, allowing flexible design of the magnetic pattern and thus the network organization.
An efficient and non-toxic magnetic nanoparticle cellular uptake is crucial. To succeed, appropriate magnetic nanoparticles are needed. The important features are their size, coating, and saturation magnetization value.
Demonstrating the procedures will be Reut, Plen, and Dafna Levenberg, master students for my lab.
To begin, cut glass slides into 2 by 2 centimeter pieces using a scriber pen. Clean the glass slides in acetone and then isopropanol for 5 minutes each in an ultrasonication bath. Then dry them with ultra-high purity nitrogen. Coat the glass with photoresist using spin coating at 6,000 rpm for 60 seconds to attain 1.5 micrometer thickness, and bake at 100 degrees Celsius for 60 seconds. Expose the sample to a light source using a photomask or maskless lithography to achieve the desired pattern. Develop the sample for 40 seconds in a developer, diluted in distilled water according to the manufacturer's instructions. Then, wash it in water for 45 seconds and dry it with UHP nitrogen gas. Inspect the pattern under an optical microscope. Insert the sample into